I will talk today about our new, more based on our research vision, what we would like to move forward in the next couple of years, or maybe 10 years, 20 years from now. So organoid intelligence, what we're thinking about, why we are thinking about that. So brain versus computer. Brain is more energy efficient. Brain has higher capacity of data storage than computer. Brain is continuously thinking and learning. Computer is faster in simple tasks, but brain is faster in complex tasks. Computer performs the task sequentially. Brain can do it sequentially and parallel processing. But computer is more affordable for us at the moment. So what we need to do, having all in mind advantages of the brain, to find the alternatives of the brain, which is a brain-like biological computers. And this is our goal, to apply organoids to do so. So in our group, we developed a model from induced pluripotent stem cells, and Thomas, who is here in the audience, Helena and David, they were main developers. The model was funded by NCATS and is licensed to Axozim. And so we started from IPS, which we differentiate into neuroprogenitors, and they putting them on a shaker. We have really nice uniformed brain organoids, and we can produce 4,000 to 6,000 brain organoids per 1 6-well plate. And if you look inside of our brain organoids, we have different cell types of central nervous system represented there. So we have different type of neurons, astrocytes, oligodendrocytes. We also recently incorporated microglia into these organoids as an immune component, which is very important not only to study neuroinflammation, but also for the neural developmental and synaptic pruning. And this is the list of different projects we have in our lab running or have been running. And I put all the QR codes of the publications we've done over the last couple of years using this brain organoid model. And you can see that it's a very versatile tool. So we can study neural development, development of neurotoxicity, neurodegeneration. My particular interest, for example, is autism and gene-environmental interaction in autism. So, but what we didn't address so far in this model, whether we can study cognition, whether we can study cell memory and learning on a cellular level, on the level of the brain organoids. And before I would like to step back and go back to the monolayer cultures. So this group in 2001 led by Steve Potter showed already that monolayer neuronal culture is able to control a robot, which you can see can move, and is entirely controlled by the just simple neurons which are sitting on multi-electrode arrays. Can you please start the video? And group in Corticals Lab, which is located in Melbourne, Australia, and we are currently collaborating with them, and you can find this Barkov paper already. They moved further and they taught their monolayer neuron cultures to play Pong. So basically, if you see this bar on the left, white bar moving, this is the cell cultures which control this bar, which is very fascinating, showing that our cell cultures are capable of memorizing simple tasks. But I'm not going into details why 3D is better than 2D. You probably heard it yesterday from John. He gave a very nice list of an overview why we need to move to 3D. And in particular, in terms of the brain and nervous system, it's more important to have very high dense cell-to-cell interaction. And we need more different cell types than only neurons. So, and this is our team who is currently working and optimizing our brain organoids model to have nice synaptogenesis. And you can see on this electron microscopy graph, the synaptic terminal. We have myelination in our model, and this is really key for electrical conductivity, is it's insulation sheet wrapping around the axons. We have 20 to 30% of the astrocytes, which is also key nutrition providers and supporter cells for the brain neurons. And as already said, we have microglia, which is extremely important for synaptic pruning. So we can look into the synaptic plasticity within these brain organoids. So how we can look into the synaptic plasticity, how we can record the electrical activity of them. And this is the schematic representation of different models which evolve over the last couple of years. It's now like a mushrooms after rain growing different options how you can record. So for example, first you can see MASH electrodes which are covered in the brain organoids. Then you can plate brain organoids on the platter multi-electrode arrays, either low density or high density. You can cover brain organoids, as you can see in this as shell. We call it brain organoid EEG. And also you can put them on a Pocky electrodes, or you can use a shunt electrode by introducing nanopixels into the brain organoids. Simple calcium imaging is also very helpful. And there is a really cool experiment showing that the electrodes which are incorporated inside the brain organoids at those wires mimicking the neuronal structures without damaging the system. So in our lab, we are trying different options. So we are working with monolayer, multi-electrode arrays, low density, high density. We have brain organoids EEG and calcium imaging. And we also have collaborators where we're about to start to use shunt electrodes and neuron-like electrodes. But before I show you our results, I want to point to this publication from our collaborator from UC San Diego, Alison Moultrie. In 2019, this hit a lot of press because what he showed with his team, that when he played his brain organoids on the simple multi-electrode array, which is low density multi-electrode array, and kept them growing for 6 months, they were continuously increasing the complexity. And what was importantly, he showed that the network level of those events he was recording on oscillation was similar to premature born oscillations in the human brain, which was obviously fascinating, showing the capability of the brain organoids. And he moved further and attached those brain organoids to the robot which they built in the lab. And the robot was also able to walk. So now back to our model. So here you can see simple calcium imaging, and probably it's a bit underestimated assay. And actually the beauty of this assay is it's really scalable. So you can do calcium imaging in high throughput and get a lot of data from it. We also recently started working closer with Maxwell Biosystem, and they have also booths here at the conference, and Daulet is working together with Cortical's lab to establish our brain organoid and Maxwell system. And it's really elegant model where you can see very tiny small chip, but it has over 26,000 electrodes. So it's really high density, and we can record on the level of single cell. And here you can see recording from one organoid. So, and you can see the different activity scan. You can see firing rate and bursting. And then here on the raster pro to the left, you can see a lot of spikes over different electrodes, and the these blue dark lines showing the burst, meaning that a lot of cells are firing at the same time. So showing the synchronicity in the electrical activity, which is, of course, this is what you want to see in your system. This is another really cool tool which Maxwell Biosystem provides on their arrays. Here we can track the electrical activity from cell soma alongside the axon. So you can see how the electrical signal is moving from the center of the cell toward the axons. And then we can see the functionality of axon and extract the axonal arbor of a single neuron or measure the velocity. And as of this set, we also have developed this EEG for brain organoids, and Chi Huan and David Gracias are the main players in this project. They are from engineers from School of Engineering at Johns Hopkins. And so they developed this very nice tool where you can have a leaves covering the brain organoids. And Chi is presenting poster today, 234. So if you would like to see more material and bioengineering part of this project, please go and visit his poster. So here you can see those cages in the first image. Here without brain organoids and the leaflets, and then those leaflets are populated with the electrodes. And then when we place the organoids and plate them in the medium, they folded around the brain organoids, and then we can record from those leaflets. So we are recording in comparison to the platter electrode array, we're recording from the surface. So EEG. And also this is the spontaneous electrical activity from these different channels. And here when we stimulate them with the glutamate, we can see increase in activity in response to the glutamate. So yeah, we can record spontaneous activity, but they have nothing to think about without input or output, right? So it's just electrical activity. So therefore, this is where AI should come into the play. And with that, we can provide input, but also output. We can analyze this big data, which we can generate from the recording after the stimulation to see whether we can train the organoids and remember their response to the stimuli. And this is from our vision is very promising way to move, and we can tackle a lot of different aspects of the neuroscience and memory studies toward organoid intelligence. So how learning changed the brain organoid, reflection of the biological concept, and how how can we do it ethically? So we ask all those questions. And of course, we cannot do it only in our lab of the Center for Alternative to Animal Testing. So we actually brought together a quite amazing group of people. And this is the core people from Johns Hopkins only. We have bioinformatician and AI specialists, we have material scientists, bioengineers, we have ethicists on board, we have retina experts on board, we have neuroscientists, electrophysiologists, big data scientists, and we have recently teamed up with Barton group, who is leading the institute in the applied physics laboratories, and they have a huge group of bioinformaticians who sitting and waiting for us to provide them with data they can play around and see whether they can find any memory or learning in our biological system. And beyond Hopkins, we also collected the interest from over 30 different people organizations, which is led also by Alison Moultrie. So we are trying to move forward a society of organoid intelligence, a community, and established organoid intelligence in this discipline. And to kick off that, we had a couple of months ago in February, we have a first workshop on organoid intelligence, and Iti, who is also present here, she was the organizer of this workshop, where we brought all those people together, from ethicists to biologists, neuroscientists, electrophysiologists, to talk about different fields. How can we bring them together, and how can we make this organoid intelligence happen? And beside that, the Dean of School of Engineering at Johns Hopkins also was very intrigued by our approach, and he put this project as a pepper line for the vision for the next 10 years for the philanthropic campaign. So what we want to do? So this is the brain organoid, how it is. We want to make it bigger. We make it more to generate more cell mass, more complexity. And we will be doing that by, and ITSI is already working on that with our bioengineering group. We are doing that with perfusion of the chemicals and performing the gradient. Then we will be recording with our shell electrode, as I already showed you, EEG. We can stimulate them simultaneously with the shunt electrodes and record also from the inside with the neural-like electrodes. And all this recording will be collected and then again analyzed by AI. And also, beside the chemical stimuli or electrical stimuli, we are planning to apply more complex stimuli, such as optogenetics, and combine brain organoids with retina, which we also have in our Hopkins in our neighbor lab available, so that we have input from the retina, which is connected to the organoid, and organoid will respond to that input. And of course, the ethics, it's really important. You cannot imagine how intensive was the discussion at our workshop when we were talking about ethics, already starting from the name. So organoid intelligence as a name is highly ethical question, and was a lot of discussion about that. And we want to have ethicists on board from very, very beginning. Therefore, Jeffrey Kahn, who is the director of Bioethics Institute in Hopkins, he's on our board and together with his team. And they are following on the concept, and they're actually also part of the NIH Brain Initiative 2.0. So we are good covered with that. Okay, so it probably sounds like a pie in the sky, and we probably don't have the biological computing yet. But with this high level goal, we can achieve many different other things on the way. We can use organoid intelligence concept to use it in medicine, to study before we can really develop in biological computing. And it's Indolet Our students, ET is postdoc on the as a PhD student, they're working toward that goal. And ET is developing the system to study neurodegeneration. So we can study the decline in the cognition in frame of the neurodegeneration, while the LET will be looking into the neural development. So for example, how we can also use different iPS donor cells from autism or Asperger and see how they perform, whether they memory and learning and memory will be different from the typically developed individuals. So we can also study dementia and side effects of drugs and so on. So there is a big room to play with. And of course, it takes a village. And I think I showed you that our village is growing, and nothing is possible without Cat family and also our collaborators. And of course, all the funding we are getting to move the field forward. And with that, thank you very much. And I open for the questions.
Hi, Randolph Ashton, University of Wisconsin-Madison. I kind of have two questions. The first is, do you have a neuronal spheroid, a cortical spheroid, or a brain organoid in your system? Neuronal spheroid being neuronal arrangement of cells with astrocytes, oligodendrocytes you can put in there. Cortical spheroid being neuronal cell types that represent all 6 layers of the cortical plate. Brain spheroid being those same cell types, but organized in a pseudolaminar structure with a ventricular zone element to it.
So we can talk a lot about the terminology here. So what we have, we don't have cortical layers, at least not what we observed. We do have different type of neurons, that's definitely. And we also have in the beginning of differentiation, it's the neural rosettes as a ventricle zones. And we consider this the system as organoid because we have myelination and active synapses, and especially when we're putting them on electrode arrays, it's a substantial model, but yes, we don't have, it's not the cortical layers, it's not as structured as other organoids which are out there. But to the major difference of this model to classical brain organoids is that they have very standardized reproducibility in terms of the size and cellular composition. So we developed that model for toxicity testing primarily. So, and that would be the key. Yeah? So we need to have a certain standardized model to be able to screen chemicals.
Thanks. The second question, I guess, in Allison's paper, he takes the brain organoid, which in this spheroidal suspension shape has the pseudolaminar structure, but has multiple sort of morphogenesis points within the organoid, which lacks biomimicry in that aspect. And then he puts it down the electrode array, and it sort of flattens out. So the electrode signatures that he is assessing and watching, quote unquote, mature are essentially neuronal networks that are maturing over age as the synapses get stronger. And the EEG signature that he is matching that to in terms of neonate is sort of measurements of the timing of the neural circuits. And he's showing that the oscillarity that they get in those structures matches in the timing. But I guess my question goes back to the idea of intelligence. And in those organoids and potentially in yours, there's critical aspects of cortical plate circuitry that are missing. And so I just, as you said, it's kind of pie in the sky, which is interesting, but I just wanted to be sort of careful on the language we use of intelligence and the idea of teaching, like the Pong exercise and stuff is sort of mapping neural circuits in a dish that stay relatively standard, and you can kind of induce synaptic strength by adding in their various excitation at certain points in that network, right? And so I guess my question is, while it is pie in the sky, do you think it's really accurate to say intelligence versus more sort of creating circuitry that is definitely biological circuitry that you can program?
Okay, so I will answer with the question, is artificial intelligence an intelligence? Is artificial intelligence an intelligence?
Again, terminology, artificial intelligence is pattern matching, to be honest.
So
And
therefore,
you can teach it to learn, and from the computational sense, you're learning patterns and sort of orienting those, and you can do deep learning and stuff like that. But those are like buzzwords, which get a lot of press, but I wouldn't necessarily say are quote unquote intelligence.
right, that is the point. So I can tell you also that organoid intelligence idea came from artificial intelligence, obviously. And also just to emphasize, we are not planning to recapitulate brain development, how it is, and learning and memory, how the neuroscientists pursuing that in vivo. Our goal is to have the level of learning and memory which those particular systems are capable of. So if they can play Pong, how can we move beyond to use that also for compute something?
One last question. There's a giant literature on the electroencephalogram inverse problem. Have you started thinking about the voxel size that you can invert to understand where and what's going on inside your brain organoids?
Yeah, it's a great question. And I think we're not there yet to start tackle these problems.